Okay. Are there any questions? Can somebody please confirm that you can hear me?
Yeah, we can hear you.
Great.
We can hear.
All right, let me get to where we're supposed to be. Let me share the screen. Nope, don't want that. All right, you should be able to see my screen now. Today is January 11th, and we're supposed to finish chapter 1 and start chapter 2. I don't think we'll get that far starting chapter 2. Let me remind me, there is a practice quiz that must be taken by Wednesday of this week. It's practice quiz 00. Let's see if I can show you the link. It's in Canvas. I don't have that up at the moment. Why don't I have that up? Any questions about that? A good reason for taking the practice quiz is it will help you learn my format, get you used to my quizzes. I will give an extra credit point for each quiz. There are actually 2 practice quizzes, one a multiple choice quiz, which is timed. There it is. Meaning you have to finish it within a certain amount of time, and you have over a minute on each question. I don't remember how many questions there are, but let's say there's around 10. And then there's a practice quiz that has essay questions. They're short answer and essay, and you have to fill them in. I have 2 quizzes because you can't put, well, they're 2 different quizzes. One is timed, the other one is not. You'll have all day Wednesday to work on the not the multiple choice questions, the essay questions. You'll have all day to work on it. But both quizzes must be done at 11:59 p.m. on Wednesday. You get 1 point each for each, 1 point extra credit each for each quiz. And where are the topics? There it is, practice quiz 00A and then practice quiz 00B. Another reason for taking the practices quiz is it will require you to download the Respondus lockdown browser to take practice quiz 00B. And that way you'll get it on your system and up and running. To take all of the multiple choice quizzes, you have to use Respondus lockdown browser. And if you download it and it doesn't run on your system, this will tell you that it's not running on your system. And so I want you to get ready before you do the quiz meaning the first quiz, quiz 1, to have Respondus lockdown browser working on your system. Because if it's not working, and about one student every other term, it doesn't work on your system, you need to contact the computer help desk and get it up and running on your system. They are experts at getting it up and running on your computer. Usually, in fact, always, the problem is that the student has a computer that has settings which are not allowing the Respondus lockdown browser to run. And I don't know what the settings are. It might be that the lockdown browser does have to change your computer so that the lockdown browser can work, and it will not allow you to open another browser or open another tab in the browser while you're taking the quiz. And so if it's not running on your system, you need to get it up and running, or you cannot take the multiple choice quizzes and exams. Any question about that? So take the practice quiz, and then you'll know that you have the lockdown browser up and running on your system. Do not do what one student in the past did. She would just delete it and then download it again, and it wouldn't work. So she would delete it and download it again, and that's not going to work. You need to contact the computer help desk, and they will walk you through changing the system on your computer to getting the lockdown browser to work. Now, most students, in fact, I think it happened last term where all the students got it to work. And so this term, all of you will get it to work except maybe one. But you need to get it up and running. So work on the practice quizzes. And you get extra credit and a little bit of a sample for what my quizzes are like. Any questions about that? You can find that under week 2 of the lecture. And there's something else I was supposed to talk about. Yeah, you can form an extra credit study group with at least one other student and quiz yourself for half an hour or longer on a material that you're going to be needing to study for the quizzes. And then submit a document, although you can type it in this format here directly. Type in the information. You need to include your full name and the full names of everyone in your study group. Tell me when you studied, just give me the date, what you studied, and how long you studied. And note, you cannot study the current week's lab assignment or work on another current assignment. Meaning you should do your lab work in your lab time. It's not for doing extra credit studying. This is an assignment that you're going to get points for, so you're not supposed to get extra credit for that. And then if everything's submitted, you'll get 1 extra credit point per week for a total of 9 points, 1 point per week. And only one person in the group needs to submit this information. Not all of you do, but you can if you want. Any questions about that? All right. And you guys, I don't have it because I'm an instructor, but you'll have a link either up here on the right or download down here to download a document, or just, I don't know, open up something. You can type that information in like a notebook pad and then submit it. And I've got the grading pad, the grading icon. All right, any questions about any of that? I think that's it. So let's move on. I think I talked about that. We'll do lab 2 today in the lab. I'll talk about that when we come to the lab. So let's go to the lesson. I should ask you, how is my background? Because I don't have my green background in front of me. I ran out of time. Boy, that doesn't look good. Val, you're seeing? Hopefully
It
you
looks
see.
nice.
Yeah, I'm not, oh, there it is. There it is there. Okay, it's fine. The background's fine. I'm not sure why I'm seeing that. Let's go and share the screen again. So we had talked about viruses last time. Any questions on last time? So when we compare the 3 domains of cellular organisms, you'll note that there are some similarities and some differences. For example, the bacteria have 1 circular chromosome, and so do the archaea. Bacteria have no histone proteins, no introns in their DNA, and eukaryotes have histones and introns. We also have generally many chromosomes. The difference is eukaryotes don't have a circular chromosome, they have a linear chromosome. A linear chromosome, like on the X chromosome, it has a starting place and an ending place. A circular chromosome is a circle, like this O right here. The beginning point is the ending point. They differ in the ribosomes. The bacteria and the archaea have 70S ribosomes. They're smaller than the 80S ribosomes. I'll talk about that later. You can find peptidoglycan in the bacteria cell wall, and generally it's not present in the archaea, and it's totally absent in the eukaryotes. Let's talk about a little bit of a brief history of microbiology. And when we're talking about the history of microbiology, the early history of microbiology was concerned with the debate over whether microbes could spontaneously generate in like a flask of media or something where the microbes could live, or whether they came about through biogenesis, meaning a microbe came about simply from another microbe. In the early history of microbiology, it was called the golden age of microbiology. You don't need to know the dates of that, but it was early in microbiology from about 1857 to 1914. It's called the golden age because whenever a microbiologist started studying something, it was something new. This slide is showing you the history of microbiology, excuse me, that we'll study, and that's showing you the golden age right there. Now, when I'm talking about the history of microbiology, I'll give you the years, but except for the first year, you don't need to know the years. Like, for example, I'm not going to ask you when were the first Babylonian clay tablets recording instructions for writing down a beer recipe. Okay, I'm not going to ask you that, but I'll give you, when I'm talking about it, that it was in 4300 BC, just to, I don't know, give you some trivial information, to give it some background information. You do need to know the first year though, and that is greater than 3.5 billion years ago. The ancestors of bacteria were found as the first living organisms on the planet Earth And you could find them in the fossil record. Right here, we're seeing an actual picture of an ancestor of bacteria about 3.5 billion years ago. And there's the artist's rendition of what the bacteria look like. Any questions about that? So the next event in the history of microbiology is that the Babylonians actually recorded recipes for making beer, and that was recorded about 4300 BC. And the Mayans did another major recording. They recorded in 600 AD a recipe for making a fermented beverage from cacao that we call chocolate today. Any question about that? Like I said, you don't need to know the years, but you should know the,
I have a comment.
okay.
In anthropology, we read about in the Amazonian how different tribes would allow their fruit to ferment in the sun, and they would have it during festivities.
Can you repeat the last part? I didn't quite hear that.
In Amazonian tribal areas, for thousands of years, they've been allowing their fruit to ferment in the sunlight, and they would have it during multitudes of their festivities.
Okay. All right. As I was saying, you do need to, thank you, you do need to know that before the Mayans wrote down their recipe for making fermented cacao, that the Babylonians were the first ones to write down a recipe, which actually was using fermentation. And then before that, you had to have the first ancestors of bacteria appear in the planet Earth. So you kind of need to know the relative order of things, but you don't need to know the dates. The next thing in the history of microbiology is in 1590, Janssen developed the first microscope, and it was a compound microscope. It was almost 100 years later, not quite, that Robert Hooke took the microscope and then saw little boxes in plant cells that we call cells. And that happened in 1665. Like I said, you don't need to know the year that Robert Hooke did that, but you should know that Robert Hooke took a microscope and looked at plants and saw cells. Okay, and he couldn't do that until obviously after Janssen developed the microscope. And then Robert Hooke's vision of cells marked the beginning of the cell theory, which is that all living things are composed of cells. Now, this cell theory was further developed by others, and Virchow's the first one who actually wrote out the cell theory and published it. But it's a theory that developed slowly over time as people looked at more and more living organisms and they saw cells. They didn't see cells in animal cells until much later than plant cells. And that's because the primitive microscopes that were around in 1665, everything looked kind of blurry. And they could see cells in plant cells because cells have a very thick cell wall, which could show up even when the cell was blurry. And then animal cells have a thinner cell membrane, which did not show up until the microscopes were developed much better. As I mentioned, the early history of microbiology saw two competing hypotheses to explain how life began. The first was spontaneous generation, and that is living things arise spontaneously in an area. A vital force comes into the region and then puts whatever is there together to generate life. The other theory was that biogenesis states that all living things come from something that was once or that is alive. For example, we say that a child comes about because of two parents. Now, over a century ago, beliefs included things like if you throw a log into the fire, sometimes you saw a newt come out of the fire. And people would say, aha, spontaneous generation of a newt. You put wood into a fire and it spontaneously generated the newt. Now, of course, the newt was just in the bark of the wood and then didn't like the temperature of the fire and so crawled out of the fire. But they didn't realize that. And they would say, aha, spontaneous generation of the newt. And then they saw flies emerging from manure and they said, aha, spontaneous generation of the flies out of manure, as well as maggots arising from decaying corpses. They were logical, but they were not scientific. And some were even amusing. Like somebody wrote in a scientific literature of the day that if you put sweaty underwear and wheat in a jar, it could give rise to mice. What happened was the mice found the wheat jar and crawled into it to get the wheat because they were hungry. And then they were simply trapped in the jar and couldn't escape. And the sweaty underwear had actually nothing to do with it. Any question about any of that?
Yeah, I missed what the two competing hypotheses were.
Spontaneous generation, that life just spontaneously generates, or biogenesis, that life arises from other life. All right, the first evidence, and actually the first scientific experiment that was ever done, was done by Francesco Redi. What he did was he got 3 jars that were either completely sealed, or he covered them with a fine wire and mesh, or he left them open. And then he put meat in the jar. In the jar that was completely open, there were no maggots that appeared in the meat. Excuse me, there were maggots that appeared in the meat of the jars that were completely open. Okay, in the jars that were completely sealed, or in the jars that were covered with a fine wire mesh net, there were no maggots that developed in the meat. And so he correctly stated that this scientific experiment does not support spontaneous generation, that maggots did not spontaneously generate from the meat. And by the way, he had a jar covered with a fine wire and mesh because when he first told people about having the jar completely sealed, they said, no, Francesco, you completely sealed it. And so the vital force could not get in and then allow spontaneous generation to come. And so he countered that by having the jar covered with a fine wire and mesh, and then the vital force could get in to the meat, and there were no maggots. So after his experiment, people would agree that for large organisms, that spontaneous generation did not occur. However, for microorganisms, you could see them growing in the meat. And so they stated that microorganisms could spontaneously generate. And there's a little bit of an explanation in the text about why that happened. Like if you got a jar and you left it open, or even if you sealed it and you put in non-sterile media, that microorganisms would grow in it. And so they would say that microbes could spontaneously generate. Any question about that? Now let's take a brief detour from that and we'll come back to it. But we need to mention that the first observations of microorganisms were observed about this time by Antony van Leeuwenhoek. And the reason why he's the first is that he's actually the best microscope builder of his day. He described seeing live microorganisms wherever he looked, like in teeth scrapings, meaning scrapings from his teeth, when he looked at rainwater, in peppercorn infusions, and then when he looked at the water from the River Thames. He saw microorganisms essentially wherever he looked. And there we're seeing a picture of Antony van Leeuwenhoek. Why he was the best microscope builder of his day was, this is actually a replica, not a replica, an actual picture of his microscope, was because he was the best lens grinder of his day. He actually ground that lens himself and then put it in his microscope that he built for the lens. And you see that pins? That's what he stuck, what he is observing under the microscope. And another reason why his microscope was better than the compound microscopes were because he only had one lens. And so it could focus sharper than two lenses put together in a compound microscope. And we'll talk more about this type of a microscope and the other microscope, which has two lenses, a compound microscope. We'll talk about that in the lab. Anyways, he was such a good microscope builder and lens grinder and such a good amateur microbiologist that he actually saw the first bacteria cells, and they were cells so small that they were not seen again in another microscope other than in his own microscopes for practically a century. And there you can see his drawings. These are looking like bacilli, and that looks like a spirillum. We'll talk about that. And then these cells here look like cocci. And these cells are so small that it took microscope builders 100 years to build a good enough microscope that other people could actually see bacteria. Any question about any of that? Now, they knew microbes existed because when you made media and you left it uncovered, it would get turbid, and that was from the growth of microorganisms inside the media. And about this time also, where do I have it? Oh, here we go. Virchow actually wrote up his theory about the cell theory that all living cells arise from pre-existing cells. Like a baby comes from two parents, a microorganism comes from a pre-existing microorganism. Okay. Anyways, that was his theory, and it's not he was the first one to propose it. It's just he's the first one who actually wrote it up and then published it. And then arguments were presented about spontaneous generation or the theory of biogenesis until Louis Pasteur in 1861 developed a theory that disproved spontaneous generation and therefore confirmed biogenesis. Now, a little bit of background. Before he did this experiment in 1861, Louis Pasteur showed that microorganisms are present in the air. And so if you have like media open to the air, microorganisms from the air can fall out of the air and contaminate the media, and then the microbes will grow in the media. Microbes, Louis Pasteur showed, could be destroyed by heat. And then he made his ingenious, famous swan neck flask experiment, where he made the media in a flask like this, and then you heat the glass right here and bent it into a swan neck. And then you have to sterilize the media, and he did that by boiling it, by subjecting it to heat. And then he found that in this tube, the microorganisms would not grow in the tube. They would, however, grow if you took the media and you brought it down here and then brought this media back, meaning that the microorganisms from the air could fall down into the flask down to here, but then with gravity, that's as far as they got. And that's why the media in the tube stayed sterile with no microbial growth. And this theory disproved the theory of spontaneous generation and then proved the theory of biogenesis. Any question about any of that? All right, I'm not hearing a question, so let's move forward. After Pasteur's work proving the disproving the theory of spontaneous generation, there was a lot of other things that came out, and people start asking the question like, do microbes cause disease? And that gave rise to the germ theory of disease. And this gave rise to the theory of immunity. And then out of both of these came the concept that we could find antimicrobial drugs, a compound that could actually inhibit antimicrobes and could be used as an antimicrobial drug. Any questions about any of that? All right. So then in the golden age of microbiology, a few other things. I'm going to say the date, but you don't need to know the date. You should just know who discovered this development and what the development was. So Louis Pasteur in 1850 developed the basic idea of aseptic technique, and that is what we use in the microbiology lab. You have to use aseptic technique with your cultures to keep them sterile or to keep them pure if they have one microorganism growing in them. Koch developed that further, and what he did was he developed a technique for streaking for isolation on solid media, and that allowed for the easy development of working with pure cultures. Louis Pasteur had a means for working with pure cultures, but it wasn't easy to get a pure culture with his means. Both Robert Koch, all you did was you streaked it out, and then you got a pure, a isolated colony, and you streaked it out again. So it went through 2 colony forming events, and then we say it is a pure culture. And then in Robert Koch's lab, Hess, a person working in Koch's lab, developed nutrient agar which was a new solid media for growing microbial growth, and we still use it today, NA or nutrient agar. It's simply a general purpose media that grows a whole lot of organisms cheaply. And then about this time, Sergei Winogradsky developed the concept that microorganisms are involved in some of the biogeochemical cycles on the planet Earth, such as the nitrogen cycle and the sulfur cycle that we've talked about earlier. And these cycles, at least some of the steps with the nitrogen cycle, most of the steps happen because microorganisms are making that step happen in the nitrogen cycle. Louis Pasteur went a little further, and in 1857, he showed that microbes are responsible for fermentation. And we'll talk about fermentation a little bit later, but let me say one of the fermentations was alcoholic fermentation, and Louis Pasteur showed that it was microbes that fermented grape juice to make wine, or fermented whatever you use to make a beer. And it was the conversion of sugar by the microbe to make alcohol, making the beer and the wine. And Louis Pasteur also showed that the microbial growth could spoil the beer or the wine or some other food as well. And so that microbial growth can spoil the food. And as you know, when you have meat out at room temperature for too long, the microbes will spoil it. And Louis Pasteur showed that what happened was, for the alcohol being spoiled, that it would convert the alcohol and produce acetic acid, which is another type of fermentation, acetic acid fermentation. Any question about any of that? All right. So Louis Pasteur showed that we could prevent the spoilage of beer and wine by microbes by heating the beer or the wine to a temperature not hot enough to evaporate the alcohol, totally evaporate the alcohol, meaning some of the alcohol would be evaporated. And this we call pasteurization, the application of a high heat for a short time, which is used in a wide variety of foods. Most of you are familiar with it for the pasteurization of milk, but those of you who drink beer, most beer is also pasteurized, the exception being Coors beer, which is sent through a filter and sterilized that way. Okay, so with the development of the theory of fermentation, that microbes can change an organic material like grape juice and ferment it into wine, beg the question, could microbes have a similar relationship with plants and animals? And may microorganisms cause disease? This concept became known as the germ theory of disease, and it was put forward by a lot of different people that microorganisms might be causing disease. You should realize that originally people believed, including scientists and doctors, that disease did not come from microorganisms, that it occurred largely from poisonous vapors that were believed to be breathed in. And then the poisonous vapors would give the disease, such as cholera. And it had a little bit of intuition, and that is when somebody was walking through a swamp, they might come down with cholera, and that was from breathing in the poisonous vapors in the swamp. Okay. In reality, they picked up the cholera organism when they went walking through the swamp. All right, you'll do that in the in-class assignment, so I'm not going to talk about it. Dr. Koch is somebody who did an experiment showing that the first microorganism was responsible for developing the first disease that was ever seen. And he proved in 1876 that the bacterium Bacillus anthracis caused the disease anthrax. And then he went through the steps that he used and published it, the experimental steps required to show that a specific microbe causes a specific disease. And this we call Koch's postulates, the steps we use to show that a given microbe causes a specific disease. With this, Robert Koch actually proved the germ theory of disease. So he proved it, but he did not propose the germ theory of disease. Everyone got that? And in 1884 is when he published his experimental steps to show that a given microbe causes a specific disease that we call the Koch's postulates. Any question about any of that? We will return to the Koch's postulates 3 more times in this class. All right, so that's an important concept. We will discuss 3 different times in this class, and you will be quizzed on it. Edward Jenner, near this time, came up with the first vaccination, and he made the first vaccine against the disease smallpox. What he did was he inoculated a person with the cowpox virus, and then the individual would come down with cowpox, and that person was protected against the disease smallpox. Now, nobody understood at the time why Edward Jenner's vaccine was protecting against smallpox. And it wasn't until Louis Pasteur did experiments with vaccines to show what was going on. And Louis Pasteur showed that what happened was that the cowpox virus was related enough to the smallpox virus so that when the immune system responded to the cowpox virus, that it also responded to the smallpox virus. And so a person would be protected from cowpox after they get the cowpox vaccine, but they were also protected against the smallpox virus with the cowpox vaccine. And that actually is where the term vaccination comes from. It's the Latin word for cow, meaning vacca is the Latin word for cow. And the protection is known as immunity. Any question about any of that? All right. So with all of this work, people suspected that it might be possible to find a drug and then use it to treat people with diseases. So you use a chemical to treat a person against a disease. And this is what chemotherapy actually is. Now, today, when we talk about chemotherapy, we usually mean a drug that is used to treat cancer. but it can be used as a drug to treat any disease, not just cancer. And when we talk about a chemotherapeutic agent, it can be used as a chemotherapeutic agent to work against an infectious disease. There are synthetic drugs, and that would be something that man makes. Carboxycillin is an example of a man-made drug. And then there are antibiotics, which the textbook defines as a chemical produced by a living organism that inhibits or kills another microbe. So penicillin G is made by a mold, and it prevents the growth of some bacteria. So it is an antibiotic, but carboxycillin is not an antibiotic because it's not produced by a living organism. It is produced by a man. And so it is a semi-synthetic drug. And that's the strict definition for an antibiotic and a synthetic drug. But understand that that's not the way that these words are used in the common language, or really they're not used that way in the clinic. And generally, when I use the term antibiotic, I will mean any drug that works against a bacteria. Okay? The first antimicrobial agent found was actually quinine from the bark of the cinchona tree that people use to treat malaria. And that was discovered sometime in the mid 1600s. And we'll talk a little bit about that later. Paul Ehrlich was the first person who found a chemotherapeutic agent that could be used to treat against a disease, which was syphilis. And in 1910, he found what he called the magic bullet, a drug that was a synthetic arsenic drug, salvarsan, that could kill bacteria. And what he did was he simply looked at different chemicals, and then after looking at about 500 different chemicals, he discovered this synthetic arsenic drug, which was workable against bacteria. Any question about any of that? All right. The first synthetic antimicrobial agent was Paul Ehrlich in 1910. The first antibiotic that was discovered was by Alexander Fleming in 1928. Like I said, you don't need to know the years. What he did was he streaked out on his plate bacteria, and he was a sloppy microbiologist, and the plate got contaminated. And then when he came back later, he left the plate at room temperature. He never put it in the incubator. And then he saw that the plate had his bacteria as well as a mold on it. But around the mold, he saw that the bacteria was not growing. And he correctly concluded that something was coming out of the penicillium mold and preventing the growth of the bacteria. And he called that an antibiotic. He was correct, and he did develop that further, but he was never able to purify the compound that was preventing the growth of the bacteria. And so although penicillin was the first antibiotic, penicillin G was the first antibiotic ever found, it was not the first antibiotic clinically used. The first antibiotic clinically used was sulfonamide, a sulfur drug that was discovered in 1935 and first clinically used, I should say, in 1935. And this was a man-made compound. Penicillin wasn't successfully tested in the clinic until the early 1940s, where it was purified and then mass produced. And it was in World War II that it first was used as an antibiotic. Any question about that? If not, let's go on and talk about microbes in a different aspect. And microbes were once classified as plants, meaning bacteria were classified as plants. And this gives rise to the term flora that is sometimes used with microbes. You might hear me use the older term microflora. That is the ancient term that you guys shouldn't use, and I'll try not to use it. You should use the current term, which we use is microbiota. The normal microbiota are the microbes that are normally present in and on a human body. And so the normal microbiota of people would be the microbes that you normally find in and on a person. Animals and plants are symbiotic to a very large microbial communities that we call their normal microbiota. And they are central to the welfare of the person or organism, the normal microbiota. And we'll talk a little bit more about that. The normal microbiota, you can find it on humans, on their skin, the oral and nasal cavities, the upper respiratory tract, the digestive tract, meaning the total gastrointestinal tract. And you can find it in the urethra, meaning the lower genitourinary tract. It's usually not found inside the organism, such internal organs and tissues like the heart, the brain. We can prevent the growth of pathogens, meaning the normal microbiota can actually prevent the growth of pathogens. It can compete for resources. So if I have a microbe on my skin, hopefully you can see I'm trying to point to my hand. The normal microbes are there and they can compete for resources or space. And so if a pathogen were to come there, it may not grow because of my normal microbes competing with it. The normal microbiota can secrete toxins that can inhibit the growth of pathogens. And we'll talk a little bit about that later. Microbes can produce nutrients and growth factors for us. For example, folic acid and vitamin K. These are two nutrients that are supplied to us by our normal microbiota in the gastrointestinal tract, meaning the lower gastrointestinal tract. And then our normal microbes help us digest our food, like E. coli in the intestine helps us digest our food. And these are all the ways that our normal microbes can help us. Any question on any of that? All right, not hearing anything. The normal microbiota do not normally cause disease. However, they can cause disease in certain circumstances. Oh, good. I'm going to finish this. They can cause disease when they're growing in regions of the body that they should not be growing. For example, if E. coli is found in the intestines, that's where it's supposed to be found. And so that's not going to cause disease, and that's okay. But if it's growing in the ear, it can cause a disease like earache, and it's not supposed to be growing there. So E. coli, when it's growing in the ear, can cause disease. Any questions about any of that? And then the normal microbiota can cause disease even when it's growing where it's supposed to be, like on the skin of a patient, like an AIDS patient, a patient that has no immune system, or a patient whose immune system is compromised. Any questions about any of that? So the normal microbiota does not normally cause disease, but under certain circumstances, it can cause disease. In all of us, if we have a stab wound, even a minor stab, like a pinprick, we could get Staphylococcus epidermidis contaminating that stab and then causing an infection. All right. Infectious diseases are diseases that result when a pathogen overcomes the host resistance. Remember, a pathogen is a microbe that causes human disease. So a pathogenic microbe is a microbe that causes disease. An infectious disease is always caused by a microbe, and that's why it's infectious. And I think we've talked about that. The other types of diseases would be an inherited disease, and that's one that's caused by genes. It's not caused by a microbe. There are environmental diseases also that are not caused by a microorganism, like somebody who plays tennis can get tennis elbow, and that's a disease, but that's caused by environmental stress on the elbow from playing tennis. An emerging infectious disease is a disease which is an infectious disease, but it's a new or changing infectious disease that is either increasing or has the potential to increase in incidence. Does anyone know a current emerging infectious disease?
COVID?
Yeah, COVID-19 is an emerging infectious disease. It's a new disease. In actuality, it's also changing, and that is we always get new variants to COVID-19. And I don't know right now if it's increasing or not, but obviously increased like over 2 years ago. It wasn't even ever in the human population. And another disease, which is, they say, an emerging infectious disease because it has the potential to increase, is the bird flu. And that's another lethal disease, or often lethal disease. And they're just worried that the bird flu could come out of birds and then be transmitted human to human. And if it does, that would be a new emerging infectious disease. At the moment, the bird flu has not evolved the capability of spreading from people to people. It only can spread from birds to people. And you have to be in very close proximity to the birds to get the disease from the birds. So it looks like for the moment, this is not going to affect people, but that could always change. So far, it hasn't for the last, I don't know, 10 or 15 years. So you need to know the definition of emerging infectious disease, a new or changing disease that is either increasing or has the potential to increase. And emerging infectious diseases are created by any category of a microorganism, including viruses and prions. There are a number of factors that can contribute to the emergence of an emerging infectious disease. I'm not going to go over them because we're a little behind schedule, and you will not be held responsible for anything in this lesson beyond slide 68. Any questions about any of that? All right. Let's move on to our next lesson. How? And that is chapter 2. Oh, not there. So it looks like we're going to we're just started. We'll only have time for a few minutes, but we'll just get started on chapter 2. That's good. We won't be as far behind as I was afraid we'd be. In chapter 2, it's on the chemical principles. And as always, I give you a slide showing you the major goals and the rough outline. Know the major goals, the terms, and we'll talk about each of these. Also know with respect to the 4 major biological molecules, be able to describe their relationships and their characteristics of monomers and polymers. And let me state that you should have had the 4 major biological molecules in a previous class, and you can get 3 of them just by looking on a cereal box or any list of the ingredients. And so what are the 4 major biological molecules? Can you name them?
Carbon, oxygen.
Proteins.
Okay, protein is correct. Carbon is not a biological molecule. That is an element. So I'm just explaining why it is not. Protein is one of the 4 major biological molecules. Can someone else give me the other 3?
Carbohydrates.
Carbohydrates.
Phosphorus.
Say again, I didn't hear that.
Lipids.
Lipids is another. And then you can find all of those on a box of cereal. The one that you don't find on a box of cereal is nucleic acids. So proteins, carbohydrates, lipids, and nucleic acids are the 4 major biological molecules. They're called this, or the molecules of life, because you only find these molecules in something that is alive or something that was once alive. So we can find these molecules like in the soil, and if you don't find them in a cell, that'll because there was once a cell in that soil that made those molecules, and then the cell died and then released them. You would never find the four major biological molecules from moon dust, for example. You should be able to describe the composition, the structure, and function of each of the four major biological molecules, and then be able to give examples of each. When we're talking about things, you should know the relative order of the scale of things. Like humans are one of the biggest things we're going to talk about. The chicken egg is the biggest cell that you're familiar with, yolk of the chicken egg. A frog egg is the next biggest cell that you're familiar with. And then a eukaryotic egg cell will be the next biggest cell, and a eukaryotic cell, the next biggest. A bacteria, the next biggest cell. Oops, didn't mean to do that. Too far. So bacteria, mycoplasmas, we might talk a little bit about that. Viruses, the next biggest thing. Complex molecules like proteins and lipids, small molecules like water, and then atoms. You should know the relative sizes of things. You do not need to know the exact size of something, okay? Although you might know that a bacteria is in the order of around 1 micrometer. And you should note that the scale has an arrow here. This isn't, and 1 micrometer, that's actually supposed to be 1 millimeter right there. You should know the sizes of things for what you can see, basically down to about the size of a frog egg. And then you need a light microscope to see. And then to see less than a light microscope, you need an electron microscope. Okay? Any question about any of that? If not, I'm going to stop here and we will. How do I put that? We will talk about this next time. And I'll see you in the lab unless there's questions. All right, I'll see you in the lab at 6:30. Bye.